during last lecture, I came up with this point, that is classification of viruses on the basis of the structure and morphology that I already covered. There are 4 different types of morphology which I will see in the viruses. The first one is helical symmetrical virus. The second one is isotypic virus. Third one is enveloped virus, and fourth one is complex or eight-tail symmetric virus. Now, when we take into consideration the nucleic acids which are present in the virions. Now, that nucleic acids which are present in the viruses, they are of mainly 2 different types. The first one is the RNA and second one is the DNA. You have studied chemistry of nucleic acids in the previous year, that is first year B.Pharm, semester 2 biochemistry. Now, in case of this, in the chemistry of nucleic acids, you have studied that there are 2 different types of nucleic acids. One is RNA and second one is DNA. Now, in RNA, ribose sugar is present. In DNA, deoxyribose sugar is present. Now, there are 4 different types of nitrogen bases. They are purines and there are 2 different types of nitrogen bases. They are purines and pyrimidines. Purines are again of 2 types. Pyrimidines are again of 2 types. In case of RNA, instead of thymine, uracil is present. Now, when you take into consideration nucleic acids, phosphoric acid moieties are also attached to these nucleic acids. Now, this entire structure is, it will be called nucleotides and nucleosides, which are present in the structure of the nucleic acids. They are present in a single stranded or double stranded form. Now, when you take into consideration single stranded and double stranded form, some type of living entities, they appear segmented genome. Now, when you take into consideration these viruses are having chemical, isohedral, complex or enveloped symmetry. Now, in case of this helical, isohedral, complex or enveloped symmetry, that symmetry is for capsid, and in the capsid, the nucleic acids are present. Now, when you take into consideration the prokaryotic cells or other living entities, the nucleus or cytoplasm is the site of replication, where the genetic material replicates. Here, in case of viruses, the site of replication is the head only. Now, when the secondary, when you take into consideration, these are the primary characteristics. In case of secondary characteristics of the viruses, four specific interactions, tissue-specific interactions have been seen, and by using that four specific interactions or tissue-specific interactions, it will be possible to characterize and classify the viruses into different types. In addition, there are the specific antigenic determinants which are present at the surfaces, that is, specific surface structures which are present on the viruses, they are also useful in the classifications. In case of viruses, some viruses get transmitted through different modes of transmissions. Some of the viruses get transmitted through the sexual transmission. Some of the viruses get transmitted through the fecal matter or urine or some other body fluids. Now, in case of viruses, mainly there are 2 main types. The first one is DNA virus, second one is RNA virus. In case of DNA virus, the genome is either double-stranded DNA or single-stranded DNA. In case of double-stranded DNA genome virus, there is an envelope structure, there is a non-envelope structure. Now, in case of non-envelope structures, see there are examples like adenovirus, adenoviridae family. There are examples like papillomaviride family. In case of envelope structure, there are examples like poxviride family, herpesviride family, and hepatoviride family. Non-enveloped viruses, one family is there that is called parvoviride family. RNA strands, that is single-stranded RNA strands, double-stranded RNA strand, single-stranded RNA strands with a positive strand, double-stranded RNA strands with a negative strand. See, positive strand wala RNAs, some viruses are non-enveloped, some are enveloped. In case of non-enveloped viruses, there are two different families. One is called Picorniaviridae family, another one is Calciviridae family. Now, under Picorniaviridae family, the poliovirus, hepatitis virus, and common cold virus are the examples. Calciviridae family में gastroenteritis is the gastroenteritis causing virus is the example. Now, in case of enveloped viruses, which are with the positive strand, RNA strand, which is single-stranded RNA, but it is positive strand, there are three different families. One is called Tobaviridae family, second one is called Flavoviridae family, third one is called Coronaviridae family, and fourth one is called Retroviridae family. Now, amongst these, the Coronaviridae family is that family, that virus, that is example SARS. It is that virus who was previously having the capacity to infect animals, and then later on they have changed the host and they have started multiplying and causing abnormalities in the human beings and animals. Among these enveloped viruses which are with positive strand, single-stranded RNA, retroviruses are the Retroviridae family is the one family which includes HIV virus. In case of Flavoviridae family, which is again single-stranded RNA with a positive strand, there are examples like yellow fever, dengue fever virus, West Nile fever, hepatitis C, Zika virus infection. In case of enveloped virus, another huge family is Tobaviridae family, which includes rubella virus and encephalitis virus. Now, among these single-stranded RNA as a genetic material with the negative cell strands, there are enveloped virus which includes Filoviridae family, Boniviridae family, Rhabdoviridae family, Orthomyxoviridae family, Paramyxoviridae family, and Arenoviridae family. Now, under Filoviridae family, Ebola virus, Marburg virus are the two examples. In case of Boniviridae family, hantavirus. The next one is hantavirus pulmonary syndrome, and one more that is called Rhabdoviridae family. Under this, rabies is the example. Orthomyxoviridae family, influenza virus. There are other examples of the Paramyxoviridae family. They are mainly mumps and measles virus. Arenoviridae family, Lassa fever. Double-stranded RNA as a genetic material which is present in the virions. They are non-enveloped viruses, and under that only one family has been included, that is called Reoviridae family. Example, gastroenteritis. Now, this is about the classification of viruses on the basis of the nucleic acids. Now, when we will take into consideration on the basis of host, there are four different types of viruses. The first one is animal virus, second one is plant virus, third one is bacteriophages, and fourth one is human virus. Now, among these then the unique example for study of viruses, unique experimental tool for studies of viruses is bacteriophages. Bacteriophages are used as the most important tool for studies of different molecular biology experiments. The reason behind that, bacteriophages are the viruses who have capacity to infect bacteria. Bacteria are the prokaryotic organisms which are cultivated easily. Bacteria are prokaryotic organisms which are easily cultivated in the laboratory by using artificial culture media. They are easily cultivated in the laboratory by using artificial culture media. Now here in the laboratory, if host is cultivated using the culture medium that is called artificial culture medium, it will be possible to allow the virion to infect that host cell which is present in the, which is cultivated in the plates, test tubes in a smaller containers. And here, after allowing the infections, the virus will grow, and that virus, after growth of the viruses, plaque formation or the pock formation will be seen in a petri plate, which indicates that on a bacterial cells the viruses have been grown. Now here, when we take into consideration the bacteriophages, the most extensively studied bacteriophages are the phages who have the capacity to infect non-motile strain of E. coli. That phages are called polyphages. Now here these polyphages as well as other types of viruses, they require living cells for their reproduction. They have the capacity to grow in a living cells. That living cells are called host cells or target cells. They are obligate intracellular parasitic organisms. They are totally dependent on the host cells for the specific constituents. Cellular energy production, initiators, precursors, synthesis is totally carried out by the host cells by using the viral genetic materials. In case of the machinery, cellular machinery for synthesis of components, some amount of components are utilized from the host cells, some amount of components are utilized from the viruses. Now after synthesis, then the viral replication will proceed. Distinctive mode of viral replication has been observed in case of viruses, namely bacteriophages. Now among these bacteriophages, some bacteriophages, they are called viruses. What they do, they have the capacity to infect bacterial cells, and when they will infect the bacterial cells, the genetic material will be incorporated in the bacterial cells. That genetic material independently will undergo replication followed by transcription and translation. Now when it will undergo independently replication followed by transcription and translation, the synthesis of viral constituent, viral components will be carried out, that is phage head synthesis, tail synthesis, tail fiber synthesis, base plate synthesis, then tail wing synthesis. Now when all these components are synthesized, then independently they will be gathered together to form the mature virion. And after a specific time interval, the delayed late gene product, that is called the enzymes who have the capacity to act on the bacterial cell wall. They will act on the bacterial cell wall, the lysis of the cells will be carried out, and the n number of viruses will be released in the surrounding environment. Such cycle of replication has been seen in a bacteriophages. They are called lytic bacteriophages or virulent bacteriophages. On the other hand, there is another cycle of replication that is called lysogenic cycle. I have not explained this in the previous lecture. Lysogenic cycle of replication. Lysogenic cycle of replication is a little bit different than the lytic cycle of replication. Now here, what happens in a lysogenic cycle of replication? In case of lysogenic cycle of replication, which is called temperature, which has been shown by temperature avirulent phages, the nucleic acid from the viral origin, when it enters in the host cell, that nucleic acid will be fused with the host genetic material, and this host genetic material, this host cell in which the viral genetic material has been incorporated, that host genetic material will undergo replication followed by transcription and translation. See, entire host genetic material, imagine, entire host genetic material, in that a specific fragment or segment of viral genetic material has been incorporated. When the host genetic material undergoes replication, the viral genetic material, viral fragment will also undergo replication, which will be followed by transcription and translation. Now when these things are observed, that cycle of replication is called lysogenic cycle of replication. Here in lysogenic cycle of replication, the viral DNA will be carried forward along with the host DNA for n number of generations. It will be carried forward for n number of generations. Now this is about the introduction, or this is about the preliminary information about the lysogenic cycle. Now here, in case of lytic cycle of replication for the bacteriophages, the most important example of bacteriophage, that is called T4 bacteriophage, who is having the capacity to show the lytic cycle or virulent lytic cycle of replication. Here that phage is called virulent phage. It is the bacteriophage. It is having the capacity to undergo the replication or multiplication, which includes 5 different steps. The first one is attachment or absorption. Second one is penetration. Third one is biosynthesis. Fourth one is biosynthesis or assembly of the phage components. Fourth one is maturation. Fifth one is release of progeny of the viruses. Release of progeny of the viruses. Now, when you will take into consideration these cycles of replication, that is called when you will take into consideration this cycle of replication, that is called lytic cycle of replication. Now that lytic cycle of replication, the first step involves, that is called attachment. Now how the phages are attached to the surfaces of the host cell? Please try to pay attention. Bacterial cell, see, you have studied the anatomy and cytology of the bacterial cell. You have studied the anatomy and cytology of the bacterial cell. Now that anatomy and cytology of the bacterial cell, when studied by you, I have given the explanation about different cell organs. That cell organ's name, most important cell organ is called cell wall, which is present in the bacterial cells as a outer membrane and inner membrane. It is present in the bacterial cell as a bilayered structure. In case of one group, that is called gram-negative bacteria, it is bilayered structure. In case of second group, that is called gram-positive bacteria, it is monolayered structure. Now, in gram-negative bacterial cell wall, which is bilayered structure, the outer membrane, inner membrane, outer membrane, inner membrane, in between them, there is a periplasmic space. Now, here, the virion, which is like this, which is having the tail fibers, tail fins, and the specific receptors which are present on the bacterial cell, that virion will be attached. Slowly, the tail fibers will go inside. Slowly, the tail fibers will go inside. The tail fibers, see, in first few hours, the tail fibers will go little bit in the outer membrane of the cell wall. Then in another few hours, they will cross the outer membrane and they will enter in the periplasmic space. After another 1 hour, they will cross the periplasmic space and they will enter in the inner membrane of the cell wall. Now, when we take into consideration the inner membrane of the cell wall, inner membrane of the cell wall is little bit rigid. They contain peptidoglycan at the specific site. As compared to the rest of the components, which are present in the cell wall, peptidoglycan is a little bit rigid and tough. It will require little bit time to allow the tail fibers to cross from the peptidoglycan. As a result, when the peptidoglycan will be crossed, see, here there are, I explained, outer membrane, periplasmic space and inner membrane. Then peptidoglycan there. When it crosses the peptidoglycan there, during that time what will happen? The tail is, which is a contractile. Tail is with a contractile sheet. So that contractile sheet will undergo slight contraction. So here what will happen? The head will be brought a little bit nearby to the cell, bacterial cell. It will cross this peptidoglycan there, then it enters in the, it crosses the inner membrane of the cell wall and then it enters in the cytoplasmic membrane. It crosses the cytoplasmic membrane also, phospholipid bilayer and then that tail fin and tail fibers will enter in the nearby to the, they crosses the cytoplasm and the tip of that enters in the cytoplasm. Now when that tip of the tail fiber or tail fin enters in the cytoplasm, the hair will be brought nearby. At that time, some pressure will be applied, some pressure will be built up or some pressure will be created in the head of the virion. And that pressure created in the head of the virion, what it will do? It will pull the nucleic acid through this tail, contractile sheath and tail in the bacterial cell. Zoonotic material will enter in the bacterial cell. Some of the viruses में क्या होता है? In that viruses, instead of only nucleic acid penetration, entire virion entire head of the virion will be penetrated inside, and then it will be subjected to the action of enzymes. The head will be degraded or envelop the structure. If it is envelop the virion, then head surrounded by envelope will come inside. Then that envelope will be destroyed by the enzymes. The head will also be destroyed. The nucleic acid will be released. Now here, attachment and penetration, these two steps are necessary. We will take into consideration. See, these are the tail fibers. These are also tail fibers. They will go little bit inside. Now, then after penetration, then the next step is multiplication, biosynthesis of Phage component. Here, to carry out multiplication or biosynthesis of cell components, first, it is essential to allow the multiplication of Phage genetic material. And for multiplication of the Phage genetic material, the first step is replication. The Phage genetic material, which is either single stranded, if it is single stranded, which is supercoiled, consider, which is little bit twisted structure. So unwinding of the strand will be carried out. If it is double stranded, again the two strands are separated. They are allowed to, or they are converted to the single stranded DNA or single stranded RNA. Now each single strand, either DNA or RNA, they will be attached with the primers and they will be attached with the enzyme which is required for the replication. Now in case of viruses which are with DNA as a genetic material, either double stranded DNA as a genetic material or single stranded DNA as a genetic material, in them DNA dependent DNA polymerase enzyme will be used for the replication of DNA. In case of viruses who are with RNA as a genetic material, that RNA is single stranded RNA or double stranded RNA. In case of these viruses, RNA dependent DNA polymerase enzyme is used for the replication, and that RNA dependent DNA polymerase is called reverse transcriptase. In case of first viruses, they are called DNA viruses. DNA dependent DNA polymerase enzyme will be used. The DNA will be replicated, double stranded DNA will be formed. From that double stranded DNA, again unwinding will take place, and using one strand, the RNA will be synthesized. That is called replication, that is called transcription. Replication followed by transcription, which will be followed by translation. Here, using the RNAs, the proteins are synthesized. Using the RNA, the proteins are synthesized. That proteins are mainly used to form the Phage head. That proteins are mainly used to form the tail. That proteins are mainly used to form tail fibers, tail fins and bristles. Now, the synthesis of these proteins will be carried out. They will be gathered together to form the specific cell organelles. Now, once these cell organelles are formed, the Phage head will be formed, which is empty. The tail will be formed, which is without contractile sheath. The tail fibers and tail fins are formed, base plates are formed. At one place, at one side, in a bacterial cell, the contractile sheath will be synthesized. Now, all these constituent, that is called head, contractile sheath, tail fibers and tail fin as well as base plate, they will be joined together. They will be joined together to form the mature virion or called bacterium. Now this is called maturation state. New Phage particles are formed. These new particles are formed they are still in a bacterial cell. Now, here, when we take into consideration these first four phases, that is attachment, penetration, biosynthesis and maturation. The replication takes place, which is followed by transcription. Replication takes place, which is followed by transcription. Now, in case of this replication and transcription, the DNA and RNA synthesis takes place. Then the translation will proceed where different types of proteins are synthesized. Now these proteins, among these proteins, some of the proteins are called immediate early proteins, some of the proteins are called delayed early proteins, some of the proteins are called immediate late proteins, while the next one is called delayed late gene products. Now these first three types of gene products, that is immediate early, delayed early and immediate late proteins, they have been formed during these three phases, 1, 2, 3. When the mature ion is synthesized, then in that case the delayed late gene products are synthesized, they are called proteins. One of the example of delayed late gene products is the enzyme that is called lysozyme, which acts on bacterial cell. Now this lysozyme will be formed, which will act on bacterial cell wall. Lysis of cell wall will be carried out and millions of viruses are released in the environment. This is called Lytic cycle of replication. This is called Lytic cycle of replication. Attachment, penetration, biosynthesis, maturation, release of the fast particles. This is called Lytic cycle of replication. Have you understood? Any doubt? Now, please try to pay attention here. If you will see previous question papers, in maximum question papers, the questions on Lytic cycle of replication before practical has been asked. So, in last almost 30 minutes, I have explained entire Lytic cycle of replication. You have to include all that in the answer. Now, next one is called lysogenic cycle of replication. Now, when you will take into consideration this lysogenic cycle of replication, this lysogenic cycle of replication is entirely different. It is entirely different than the Lytic cycle of replication. Then it is entirely different than the Lytic cycle of replication. In a lysogenic cycle of replication, what happens? Phage genome, which is the genetic material of the phage, that phage genome or bacteriophage will follow phage genome before that phage genome. The bacteriophage, what it will do? It will follow the first two steps in a similar way like Lytic cycle of replication, that is called attachment and penetration. Now, when we will take into consideration the example of lysogenic cycle of replication, lambda bacteriophage is having the capacity to undergo lysogenic cycle of replication. Instead of carrying out the lysis of the host cell, instead of carrying out the killing of the host cell, here this bacteriophage called lambda bacteriophage, what it will do? It will incorporate the phage genome, or it will incorporate the phage genetic material. It will incorporate the phage genetic material into the bacterial chromosome. Now, see here, this is the first and the most important differentiating feature with the lytic cycle of replication. That is, in a lytic cycle or virulent phages where the replication takes place. In that case, what happens? The phage genome, which is incorporated in the host cell, it will undergo the biosynthesis, that is multiplication, independently. It will undergo the replication followed by transcription and translation. The proteins are formed and the new phage will be synthesized. In case of Lambda-bacteriophage, what happens? The genetic material of the phage will be incorporated or integrated into the bacteria, into the host, that is bacteria. And when it will be integrated in the host, the resulting structure is called a prophage. The integrated phage genome in a bacterial cell, that is called a prophage. Now, this prophage is having another name that is called lysogen. And by this prophage or lysogen, the multiplication will be carried out, that is called lysogenic cycle of multiplication. Here, this lysogenic cycle of replication has been observed in a temperate phage, or it has been observed in a latent or inactive cells where the bacterium replicates its chromosome, and when it replicates its chromosome along with the bacterial DNA, the viral DNA will also be replicated. After replication of bacterial DNA and viral DNA, the specific proteins are produced from the bacterial origin. That proteins will be assembled together to form new bacteria. So here, in a daughter cell, along with the other constituents, the prophage DNA will also be brought. That is called the lysogenic cycle of replication. Here, the lysogenic cycle of replication has been seen in different bacteria. The most important phase in a lysogenic cycle of replication is some of the viruses, what they do, they have the capacity to change the phenotypic characteristics in host. And such a thing, when it will be implemented, that is called phage conversion or lysogenic conversions. Now these phage conversions or lysogenic conversions have been observed in the bacteria which are pathogenic. They are called Vibrio cholerae and Clostridium botulinum. Here in this case, the phage infects the bacterial cells. The phages are having the capacity to carry the genes responsible for the toxin generation. That is, the genome which is responsible for enhancement of virulent characteristics have been hidden in a phage genome, in a Vibrio cholerae phage, or it has been hidden in a Clostridium botulinum. Now, in case of Clostridium botulinum, what will happen? Clostridium botulinum organism में phage is there. That phage में toxin generating gene is there. That toxin generating gene will produce the toxin, and because of that, the abnormalities will be seen. In case of lysogeny, here it will be possible for that lysogenic phage to undergo inductions, and that inductions are carried out in a viral genome. In case of Lytic cycle as well as in case of Lytic cycle, the virions are synthesized newly, but here in case of lysogenic cycle, the virions are not synthesized newly, but the genetic material of the virion will be carried forward for the series of generations. See, there are two important proteins. One is repressor protein, another one is repressor protein or pro-protein. These are pro genes and repressor genes. What they will do? They will try to block the Lytic cycle of replication, and they will try to operate the lysogenic cycle of replication. Here, in case of bacterial cells, for a series of generations, the viral DNA will be carried forward. See, consider from 4th generation up to 20th generation, the viral DNA will be forwarded. At 21st generation, the specific type of proteins will be synthesized. The gene which is responsible for the synthesis of proteins will get activated, and after activation of that gene, that proteins are synthesized. What will happen? After that, in the next generation, the viral genetic material will not be integrated in the bacterial genetic material, but that viral genetic material will be kept free. And when that viral genetic material will be kept free, 21st generation may, the viral genetic material will be kept free outside. It is not allowed to integrate in the host genetic material, and then it will not be allowed to integrate in the host genetic material. Then that genetic material will independently synthesize the fast head, tail, and tail fins and tail fibers as well as base tail. And the mature virion will be released. So at 21st generation, lysogeny will be stopped and Lytic cycle will be carried out. That is again the unique example. See, there are different types. These are called transductions. There are two types of transductions: specialized transductions and abortive transductions, which have been seen in the viruses. And switching of the lysogenic cycle and switching on of the Lytic cycle will be carried out in the cells. Have you understood this point? Yes, okay. Next point is cultivation of the viruses. Now, when you will take into consideration cultivation of the viruses among your brain, see, in today's lecture, what I have done, I told you the classification of viruses on the basis of the genetic material, that is DNA viruses and RNA viruses. Then I told about the bacteriophages. The most extensively studied bacteriophages are polyphages, who have the capacity to infect non-motile strain of E. coli. Now after that, I told about the virulent phages, or Lytic phages. Then I told about the avirulent phages or temperate phages or the lysogenic phages. Then I turned towards the Lytic cycle of replication. I have completed Lytic cycle of replication. Then I turned towards lysogenic cycle of replication. Now, when you will take into consideration these things, from the polyphages, the most extensively studied viruses, that is bacteriophages, are polyphages. From that point onwards, the explanation is totally restricted to bacteria. Explanation is totally restricted to bacteria and phages, that is bacteriophages. In case of next point, that is called cultivation of cells. Here I will try to tell you about the cultivation of bacteriophages, and then I will switch on to the cultivation of animal viruses and plant viruses. Sorry, I will tell you the cultivation of bacteriophages, then I will switch on to the plant viruses, and then I will switch on to the animal viruses. Now when we will take into consideration the cultivation of bacteriophages, to carry out cultivation of bacteriophages, it is possible to do the cultivations in the laboratory. For that, the media which are selected for one particular bacterium, which is the host for the phage, that medium will be used. On that media, suppose polyphages you want to isolate, then in that case, first you have to select the medium. That medium is normal laboratory medium, consider beauty and agar or Muller interagar medium. That medium, beauty and agar medium, Muller interagar, beauty and agar medium or Mekong agar medium will be selected. Consider Mekong agar is the differential medium, but on Mekong agar, the growth of the organism, that is E. coli, is observed in the form of red colored colony. So either use beauty and agar ordinary medium or Mekong agar medium. That media are prepared which are transferred in a aliquots of quantity to the petri plate. Allow that to solidify. After solidification, using suitable technique, that is by using soft agar or by using the aliquots of culture and spreading technique, the bacterial cell or mixture of bacterial cells is cultivated, is added on the basal medium plate. Now please pay attention. Now here, normally for this technique, allow for addition of bacterial cells to the basal medium, solidified medium. Normally instead of spray plate method, the soft agar technique is used. Here, when you will overlay the 10 ml or 15 ml of the medium with the soft agar, to which the suspensions or mixture has been added, it will uniformly spread all over the surface. Then to this, you have to add polyphages. Now what is the source of polyphages? Domestic waste or sewage, which is treated with a chloroform, centrifugations are carried out, some other chemical agents are also added, and the phage suspensions are prepared. They are from the sewage unit. That phage suspensions are added in a phage block. Or here, instead of using phage block, treat the sewage. Another method is treat the sewage directly with a chloroform, centrifuge it. The supernatant will be collected. That supernatant is containing the phages. Aliquots of that supernatant is added in a saline or it is added in a phage block. And that phage block, see, instead of saline, it is a phage block is better. Then that phage block will be added in, consider 200 microliter quantity or 100 microliter quantity on the surface of that medium which is overlaid with the test organism by using the soft agar technique. Then allow that plate to move like this so that fast broth will be uniformly spread. Then incubate the plates at 27 degrees Celsius temperature for 24 to 48 hours. Please try to pay attention. Here, when you are using soft agar and when you are overlaying that soft agar on the basal medium, on that when you will add the fast broth suspensions, the plate should not be kept in an inverted manner in the incubator. The reason behind that, if soft agar में little bit lesser concentration of agar is taken, or the fast broth, if you have taken a little bit larger quantity, then that will come in a lead if you will keep it inverted. Keep it in the ordinary way. The bacterial cell will grow. Please try to pay attention. During incubation, what will happen? Bacterial cells which are from the soft agar, what they will do? They will take the nutrients from the medium, they will grow in the form of coloniform. Now, if you have sprayed the fast suspension, suppose hundreds of phages are sprayed on the surface of the medium. Now, then what that phage will do? The cell will grow. That cell, after its growth, the colonies will be formed. That colonies will be lysed. Lysis of colonies will be carried out by the virus. And virus will grow on in the form of plaques or pox on the surface of the medium. When that plaques or pox are seen, you will come to the conclusion कि bacterial cell में virus का growth is there, which is seen in the form of plaques or pox. That plaques or pox are separated out, and they will be treated with the chloroform and centrifugation and some other ways of treatment, and from that the phages have been observed. This is the cultivation of bacteriophages in the laboratory. Is that clear to you? Now the next one is cultivation of plant viruses. To carry out cultivation of this plant virus का cultivation is again 4 to 5 lines का explanation. This is, it requires minimum half an hour. So I will explain that in the next lecture. Plant virus I will try to explain to you. Plant virus का cultivation. To carry out cultivation of the plant viruses, here single cell suspensions of the plant cells is allowed to cultivate in the medium. For plant cultures in the laboratory, tissue culture media are there, plant tissue culture media are there. Using that tissue culture medium, leaf of the plant, stem of the plant, flowers of the plant, from that a single cell line suspensions are obtained, and they are allowed to cultivate in that culture medium. Now here, in a culture medium, the plant cells will grow in the form of monolayer, single layer. Cells will grow in the form of monolayer, that is called single layer. Now on this monolayer culture, the viruses are allowed to multiply. Using monolayer cell lines of the plant cells, the viruses are cultivated. This is about plant virus cultivation. Bacterial viruses for cultivation I have already told. Now the next point is cultivation of animal viruses. That cultivation of animal viruses